Hello, good morning.
Morning, Professor.
How are you?
Good, Professor. Good.
Mhm.
So we need some more time. I think the exam is coming.
Yeah, okay. When you like, you can just tell me, okay? I will be there.
Sure, Professor.
Okay, thank you.
So shall we start then? I think we have given enough time for the students to Professor.
Yeah. Sorry.
Then we can resume the day 4 of GIAN lecture. I invite Professor Sena to share the screen and request you to give the lecture. Thank you.
No, this one. Can you see my screen?
Yes, Professor.
Yeah.
Yeah, I can see.
Good. Okay. So good morning to everyone, and thank you again to be with us. Tomorrow is the last day. So today we will just finish. I would like just to give you some more examples of marine-based polymers, and then I will send you. I will show you again a video about this kind of application of these marine-based polymers. And in the last part of our lecture today, I will define what is blue biotechnology, some definitions about blue biotechnology, but also blue bioeconomy and biorefinery. And at the end, I will show you I will present you my research group. So what we are doing here, and it will be maybe an opportunity to you to see what we are doing and to do some works in collaboration if you like, or to ask for some fellowships together. Okay, so please don't hesitate to send me an email or to contact me. Okay, so this is the outline for today. Oh yes, and then you will have the tutorial. So I sent to you some video. And in this list there are also the video that we saw yesterday, sorry. And you will also have a link about, so it's calls Ask Nature, and this tutorial, it's it calls Nature equal to future, and it can help you for our last lecture tomorrow, that it's about bio-inspired materials. Okay, So here you have the learning outcomes. So to identify marine-based chemical structures and the properties, and also some notions about biotechnology. Okay. So the polymer, so there are three polymers, two from animal, they are both both from marine bioresources. So I have chitosan and collagen, and then finally from algae I have alginate. Okay, so starting by chitosan, we already discussed a little bit about chitosan in the other lectures, and here I have only, so as I told you, Henri Bracano that discovered chitin. And then I have also here the people that were responsible for the discovery of chitosan and for their chemical structure. Here it's a little bit about the importance of chitosan in academic research. So as you can see here, so this is from 2019, but it's still increasing the interest to chitosan because as maybe you already know, it's the interest. It's fantastic because chitosan we can also give to this polysaccharide different forms of materials, and it's really very interesting because has very good biological properties and then can be used easily in biomedical applications. How to have chitosan? So as I already told you, chitosan it's the main derivative of chitin. And actually, we need only to do deacetylation synthesis, and we can, or a reaction, and we can transform chitin. So in chitosan, and as you can see in carbon 2, we have this amino group. We can do it by chemical deacetylation, but also enzymatic. Okay, so you can see that the conditions that we need to work, they are really very different. But also, as I told you, so to take care about this chitosan, if it's really by a biopolymer or not. So if we consider that it's directly extracted from mushrooms, so we can consider that it's a biopolymer, and it's a biopolymer. But just pay attention when we are talking about chitosan that we obtain from chitin. So what we really are extracting from nature, it's chitin. But here in the mushrooms, you can also to extract chitosan. And I know that there are a company or two in Spain that they are already producing chitosan. And also here in France is true, chitosan from mushrooms. So the properties of chitosan, as I told you, it's really very interesting because as you can see, it's non-toxic, biocompatible, biodegradable, and renewable. It's odorless, and what we need to know, it's again, as all of these biopolymers, the thing is depends of the source that we are extracting. And and this extraction method, it's also depending of it, we can have physical, chemical, and biological properties that are different. Okay. And for the extraction method, for sure that the temperature, the concentration of the solvent that you are using, or the reactive, the time, it can influence it. And I have here just a small image that show it, because depending on when we will do the extraction of chitin or chitosan from mushrooms, and then the chemical reaction, it will for sure modify the degree of acetylation or deacetylation, the molecular weight of our polymer, and also the pattern of deacetylation, okay? And you can see here that we can have different patterns of deacetylation. Still in the properties, so as chitosan has these amino groups, it means that has a great property, so to absorb the metallic ions in at the surface of the chitosan backbone, and for sure that more deacetylated it's our chitosan, more places that we have, or more groups NH2 we have, and the chelation it's better. And for sure, the degree of acetylation will also influence the solubility of the chitosan. And you remember yesterday when we were discussing about chitin, so the degree of acetylation of chitin, I told you, if it's more than 50%, it means that it's chitin. So another way that you have to know if it's chitin or chitosan, for instance, if you can't do, if you are not able to see the degree of the, acetylation, so what we can do is to take your powder and put it in a very in a acetic acid solution, for instance, and if it's if you can soluble your powder, that means that it's chitosan, and if you are not able to dissolve it, it means that it's chitin. And for sure that this percentage of acetylation or deacetylation will influence the dissolution of chitosan. So this is very important. And another properties or other properties that can be influenced by the degree of acetylation, so or deacetylation, so the percentage of amine groups that you have in our chitosan backbone. So this will for sure have an influence on the microbial, antifungal, and antioxidant properties, because it's these NH2 groups that give these properties to our molecule. So these are really very, it's a property that it's very important. So you have the molecular weight for sure, because it will influence the properties of your final material, but also the degree of acetylation or deacetylation. It's also important to know about chitosan. And for instance, in the European Association of chitin that I told you yesterday, I know that there are some people working on how to mention the properties of chitosan that you have in the paper. For instance, you need to put the source of your chitosan, the molecular weight, the degree of acetylation. This is really very important because it's influenciate the properties of your material. And if you want to compare, you need to know it, okay? And I know that it's very important, and there are some reviewers that they refuse the paper if you don't have this kind of information. For instance, I have chitosan here in the lab that it's from India, from a company from India, and it's very pure chitosan. It's for medical applications, and every time that we receive a sample, we characterize our chitosan. And I have another student that as he is doing nanoparticles of chitosan with different chitosan sources, he also take chitosan from Sigma. And he told me, oh, Susanna, we don't need to characterize it because we have already the information from Sigma. But no, because in Sigma, it gives you a range. So it's better that you characterize your chitosan and then you know really what kind of chitosan you have, okay? So this is something that it's really very important about chitosan. So here, for chitosan, so you remember for chitin and cellulose and starch. So we have different OH groups. So for cellulose and starch, we have, for instance, 3 OH groups in carbon 2, 4, and 6. And here in chitosan, so we will have 2 hydroxyl groups that we can do some modifications, but also the amino group that it's very easy then to modify with the carboxylic acid, for instance, groups. So this is very easy to modify. And here I have some kind of modifications that you can do with chitosan. Okay. And yesterday, there are someone that asked me about cancer and about the cancer treatments and the biopolymers. So I was looking and I found this review. It's really very nice. It's about nano theranostics regarding chitosan. So I invite you to see this review because you have the kind of applications that you can do, but you also have all the kind of modifications that you can do with chitosan and how to prepare this nano theranostic. So it's really a nice publication. So for processing chitosan, as chitosan is soluble in weak acid, acetic acid, for instance, in vinegar, if you like, you can also to dissolve chitosan. You can have this solution, and then you can prepare different kind of materials. So microspheres, nanospheres, nanoparticles, films, by casting, evaporation, composites, scaffolds, 3D photo scaffolds. So you can imagine a lot of kind of materials with chitosan, and this is really very coatings for sure. This is really very nice. Hydrogels. There are also a lot of research regarding hydrogels for contact lens, etc. So it's very easy to processing this polysaccharide. Applications. So I know that some of you are also interested about water and waste treatments. So I have here this in this publication. I don't work directly in this kind of applications for water treatment, but I had a collaboration 5 years ago or 6 years ago, and we used very simple way to remove some inks and metallic ions from water. And for instance, we used chitosan powder with different acetylations or deacetylations degree, and also nanochitin particles, and it was really working. And at this moment, we are just finishing a paper about the use of hydrogels from chitosan. It was a student, Unimaya, from your university, from Matani University, that she did a master degree with us, and she started to work, and now I have a postdoc that it's finishing the work, and it's about hydrogels for water treatments with the inks in our case. So you have this kind of applications in agriculture, also as bio nutrient or a bio because of the nitrogen, or a biopesticide. It's also been used to do it. Food, cosmetic, biopharmaceuticals, but for sure in biomedical application, it's really, really very used. So let's move now for the other no, in this case, it's not a polysaccharide, it's a biopolymer, collagen, so, but it's a protein. And so collagen, I think that I show you this image to you the first day when I present you the proteins in general. So collagen is the most essential extracellular protein that we have in animal body. And it's involved in the mechanical protection of tissue and organs. So our skin, for instance, our bodies, we have collagen everywhere. And I show you here this multi-scale hierarchy of collagen fibers like chitin and silk. And so these four biofibers, so cellulose, chitin, silk, and collagen, they in nature they are in the form of fibers. And so we have the amino acids that assemble together to do collagen molecules, and then collagen fibrils, and finally the collagen fibers. And as I told you, so the main amino acids that we have in collagen, it's glycine and the proline, but we also have some hydroxyproline. So I have here the image of these three amino acids. So where we have collagen in our body, cartilage, bone, skin, cognitive tissue, hair, and the nails, and the sources of collagen. So there are, as you can see here in the left side, there are a lot of sources of collagen. So we can also do recombinant collagen, so bovine, porcine, also for mouses, human. So during long time, actually, we remove collagen from our body from one place to replace where we we need to replace something in our body. And recently, so we also have marine origin. The advantages of marine sources, it's because we can have it from wastes. For instance, we can extract collagen. Let me see, just, okay, yes, I have then an example. But so this kind of source, we have high collagen content, no risk of disease. And then there are also some ethical conflicts that some people wouldn't like to have a scaffold or a drug or something that it's from animal or from porcine or bovine origin. So we also have low inflammatory response and less amount of biological toxins and contaminants with marine origin. So now, yes, I will tell you some marine origins of collagen. We have, for instance, shark, the starfish. I know a laboratory here in France that it's working on the production of collagen from a starfish or extraction, because in some regions, starfish, for instance, in Portugal, I know that in my region, it's not allowed to capture starfish. But for instance, here in France, there are a region that the starfish, it's a problem for local fisheries or local fishing or fishermen. And so they can capture some tons of starfish to valorize the collagen. The ones that they are coming in the net of the fishing nets. And jellyfish. Here in the lab, we are working with collagen from jellyfish. It's from a company in UK, and then from fisheries wastes. So we are extracting collagen from skin, fish, and from scales. And usually collagen, we can extract collagen via chemical or enzymatic reactions. So we can remove collagen, but usually what we have, because it's a very strong acid hydrolysis, we have gelatin. and then we can use this gelatin in different kind of applications. And we can also have gelatin, but also collagen peptides. And there are a lot of research using peptides. I'll also show you some examples in the first day. And then I have here, this is a very good review from Marine Drugs. So they also talk in this review about the biomedical applications for biomaterials, tissue engineering, regenerative medicine, dental applications, wood dressing, and also for cosmetic and drug delivery. Here in the lab, we are interested for maxillar bone, the research that we are doing with jellyfish and collagen and scales collagen. It's for tissue engineering. So the extraction, I have here just some examples of chemical extraction. So acid soluble collagen. So by using acid solution, we can also use some salt soluble collagen. So we can use, for instance, sodium chloride solution to extract collagen, and also pepsin, okay? Regarding the physical mechanical extraction, so the most used one is ultrasound. So that we can obtain the collagen to use that, to use it then to do some chemical modifications and materials. So why are we interested by collagen? It's because of this biological activity or properties. So again, we can use gelatin or peptides. So it has anti-tumor activities. It also helps cell proliferation activity. For instance, with the work that we have here, that we are studying or trying to do scaffolds for maxilla, we are using chitosan and collagen. And what I can tell you is that yes, we have the cells proliferation. It's better using collagen than chitosan, because collagen, it's really the molecule that we have in our body and that the cells they like. So we have really better results with this. Anti-aging activity. So I don't know for you, but here in France now it's like all people want to take some collagen as food supplementary because of anti-aging activity or antioxidant. So everywhere you have collagen capsules, collagen fibers, or powder that you can take it. I prefer to eat skin fish, for instance. I like skin fish, some of them. And yes, it's the way that, yeah, I'm joking. Okay. So collagen forms that we can do. So as I show you before with chitosan, so you can have 3D foams or scaffolds, as you can see, fibers, films. There are some groups that are also working. Oh, I forgot to put the source of this image in 3D printing using collagen. We can also do extrusion fibers with collagen and tubular films. Okay. So in general, the most applications of collagen again. And yeah, as I told you, for as a supplement, so help improve skin and hair. So you can, it's very easy now to buy collagen peptides for cosmetic and the nutritional activity. And here I have a lot of references about collagen sources, applications, and extraction, if you are interested in it. And now we will move for the last marine-based polysaccharide again. Now we return to polysaccharide, so alginate. So here I have a little bit of story of alginate, the discovery of alginate, and the person that discovered alginate from algae. Why, or the importance of alginates? I found this it's also a review. It's very nice if you like this polymer or biopolymer. And actually, I put all the figures that summary the properties and the importance of alginate in the presentation I take from this paper. The importance of alginate, it's a stable form of polysaccharide. We have a lot of applications that then I will show you some of them. It's biodegradable, biocompatible, also have bioadhesive properties, and is for sure from natural origin. And also, like a lot of polymers or biopolymers or polysaccharides that I show you, also have very good gelation properties. So the origin of alginate. So here I show you as marine origin, because we are talking about marine biopolymers, but we can also to extract or to biosynthesize, or so there are some bacteria that are biosynthesizing alginate, and we can do it by the biotechnological way, by extracting from bacteria. But the marine origin, it's a brown algae, and I have here, from this publication, the main algae that we can extract alginate. Okay, regarding the structure, so it's a linear anionic polysaccharide, and it's very interesting because it's a block-like structure. I don't know if you remember your polymer or physicochemical polymer materials class. Maybe you know about it, what it's a block polymer or copolymer, you remember. So you have different blocks of monomers. So here you have, for instance, the G block. So it's the block that we have hyaluronic acid, and then we have the M block, where the monomers are mannuronic acid. So you see, you have G block, M block, and depending of the source, the way that you are extracting, etc. So you will have this different size of blocks, and so you have blocks, but then you also have GM block. It's really very, very interesting, that give interesting properties to alginate. And the length of for the properties of the alginate, for sure, the length of the G block, it's important, and also the ratio of Mg block, the molecular mass or weight, for sure, will influence the physical chemical properties of this polysaccharide. So now regarding the applications, medicine, cosmetic, and food, you see the polysaccharides, the main domains or sectors, they are always the same, and then could change regarding the properties. So again, tissue engineering, wound healing, dressing, drug delivery. In terms of cosmetic, so as emulsifier, preservative, using in food industry, so as a food coating agent, stabilizer, food processing agent, etc. So it's thickeners for sure. It's we can have again a lot of kind of applications using this kind of polysaccharides. Okay, so just before to go to the other part of our lecture, I would like to show you again a film about the use of marine polymers. Can you see it? Can you see?
Yes,
Nature du futur. Des vaisseaux marins. Les algues constituent une part
Sorry.
très importante.
I start again. I forgot to put
Nature égale futur. Des vaisseaux marins. Les algues constituent une part très importante de la biodiversité marine. Ce sont des organismes qui ont la particularité d'être flexibles et malléables tout en étant compacts et solides. Ces propriétés sont dues à leur composition moléculaire particulière, constituée de longues chaînes de molécules qu'on appelle des polysaccharides. Ces polysaccharides marins sont biocompatibles. Ils pourraient donc servir de matériaux de substitution pour le corps humain.
Alors, le but de nos recherches, c'est d'essayer de trouver une solution aux vaisseaux sanguins, donc aux veines et aux artères, pour des remplacements lorsqu'il y a des pathologies cardiovasculaires. Donc, on produit des gros vaisseaux, mais pas les... On s'est spécialisé là-dessus. La difficulté principale, c'est que lorsqu'on va implanter ce vaisseau dans le corps humain, il va subir des variations de pression. Et donc, ce vaisseau doit encaisser, pour parler simplement, ces variations de pression. Donc, il doit pouvoir s'étirer sans éclater. On a beaucoup travaillé sur les plantes et les animaux terrestres pour trouver des propriétés ou des médicaments. Et on s'est dit quand même que l'océan était une partie immense de notre Terre et qu'il y avait certainement des éléments intéressants à les chercher, donc dans les algues marines et les animaux marins. Parmi les polysaccharides qu'on a trouvés, on a l'alginate, le carragénane, la chitine. Ce sont des épaississants, des gélifiants. Et donc, nous, chimiquement, on va pouvoir utiliser ces propriétés de gélification pour former des vaisseaux artificiels. Donc, nous prenons les polysaccharides, nous les solubilisons, nous les transformons en hydrogel pour avoir un aspect un peu plus visqueux. Ensuite, nous utilisons des moules pour pouvoir former ces hydrogels en forme d'un vaisseau. Nous les séchons. Et ensuite, nous les réhydratons et nous étudions leur élasticité.
Ces vaisseaux artificiels ont des caractéristiques uniques de souplesse et de résistance grâce aux propriétés de flexibilité, de malléabilité et de solidité des polysaccharides marins qui les composent.
Les vaisseaux artificiels doivent être déjà biocompatibles, donc pouvoir être mis dans le corps humain sans générer de réactions de rejet et avoir cette capacité à pouvoir s'étirer en fonction des variations de pression pour ne pas exploser et aussi permettre au sang de circuler sans faire de cailloux et surtout de ne pas fuir parce qu'il va falloir les connecter aux vaisseaux existants. Pour valider donc la fonctionnalité de notre futur vaisseau artificiel sanguin, nous allons faire un modèle animal. Lorsqu'on implante notre vaisseau dans le rat, en lieu et place de la partie d'aorte qu'on a enlevée, c'est un biomatériau qui n'a rien à voir avec la structure d'une artère, qui est faite de plusieurs tuniques et notamment de cellules endothéliales. La structure de notre vaisseau est telle qu'il y a de la porosité. Cette porosité, des petits trous, vont permettre aux cellules du rat de coloniser ce matériau et au cours du temps de le transformer en un véritable vaisseau sanguin. Les pathologies que nous souhaitons traiter avec ces vaisseaux sanguins artificiels sont bien entendu les pathologies cardiovasculaires. Lorsque les personnes font des thrombies, c'est-à-dire qu'une obstruction de leurs artères ou vaisseaux, plutôt que prélever des veines ou des artères pour remplacer ces vaisseaux malades, nous utiliserons nos vaisseaux artificiels. Sur les 3/4 des vaisseaux que nous avons implantés, tous les animaux ont survécu. La nature a encore plein de choses à nous apporter. On n'en est qu'au tout début de l'étude des plantes aquatiques et des animaux aquatiques.
Les polysaccharides issus des algues et des animaux marins constituent des matériaux nouveaux. Ils peuvent aussi servir à réaliser des nanoparticules afin de mieux traiter les maladies cardiovasculaires.
Okay. Sorry. Okay. So you see, actually, can you hear me? It's okay?
Yes, Professor, we can hear.
Okay. Oh, sorry. Okay, so it was to show you a medical application. So the things that you see, it was with alginate and the chitin. So you see, it was just to show you a way, an application about this important polysaccharides, or the same for collagen as a protein. Okay. Why I show you a lot of things about marine, because as you will see now, I work more with everything, and as I told you already, with marine biopolymers. Okay. Okay, so let's start now the second part of the lecture about blue biotechnology. And as I told you before, first I will tell you some definitions about what is blue biotechnology. And then I have again a video, again to show you an example with marine again. And then I will show you just a few slides about my research group. Okay. Okay. So blue biotechnology. So this the definitions that I have here, I see. So it's the definitions from the European Union that we have, because we have a lot of restrictions and laws that we need to know when we are doing research, because it's our responsibility also what we are doing. So these are the definitions that if you go to the EU web page, you will find the same definitions. Okay, so you can tell that it's blue biotechnology or marine biotechnology. Blue because water or the ocean, we see it blue, so we call blue. But it's not the blue biotechnology. It's not only for marine environment, it's for all aquatic environments. So when we are talking about something from rivers, lakes, it means that it's also blue biotechnology. Okay, so biotechnology means any technological applications that use biological systems, living organisms, or their derivatives to make or modify products or process for a specific use. Okay, so the process that transform marine or aquatic resources into service and goods in a multitude of fields. So again, we can use it for industry, energy, agriculture, or health. Okay, so what includes the biological, the living organisms? So regarding the living organisms, includes everything. So microorganisms like some of this image that I have here: micro, algae, bacteria, and fungi, plants, algae, and animals, invertebrates, and also vertebrates. Okay, so everything that we transform in a service or in a product, so from marine or aquatic environments, it means that we are doing blue biotechnology. And here I found again this very schematic image about it. So we have the marine living resources, and but it's not only. So as I told you here, it's biological systems and living organisms. Okay, because we can use the organisms. So if there are wastes, we can use it to valorize it, but we can also see the way that the nature or the system they communicate, or they do some management between between them. So we can use this marine ecosystems as model. So ecosystem management for services, ecosystem management. So these are things that, so how to manage some aquatic systems, or the laws that we do about it to protect the biodiversity, for instance, how to do or how to capture these living organisms. So it's what we call ecosystem management. We do research, like for instance, some of you, me, and bioremediation also. And then it's what we also call, yes, aquaculture. So what is here as culture is aquaculture. It's also included in this blue biotechnology. And then we can do some products. So drugs, biofuel, electricity, for instance, with the energy of the waves. We can do energy, food, enzymes. We can extract enzymes, bacteria, and then enzymes from marine ecosystems, compounds, DNA, blueprints, etc. Okay, in terms of biotechnology, so it's the biodiscovery, molecular biology, biochemistry, nanotechnology, bioinformatics, omics, etc. Okay, so this is just to have this panorama of the blue biotechnology. So when we talk about blue biotechnology or technology, we can also think about bioeconomy. So there is still non-consensus about the definition of bioeconomy. Again, so I see this one that is recommended by the Organization of Economic Cooperation and Development, that is the set of economic activities related to the innovation, development, and production of biological products and process. Okay, and the blue bioeconomy, we can so to have a definition like comprise the use of renewable biological resources from terrestrial. Why terrestrial? Because the plants and all the ecosystem that's near to the marine ecosystem, so the beach and these kind of places, they are also considered as marine, and aquatic systems and marine bioresources. Okay, so these are the definitions. Circular economy. So we see in the first day that I put you the linear economy, so how, or the linear production of synthetic polymers and the circular production of biopolymers. So here it's the same thing. So the circular economy, it's the economic concept that fits into the framework of sustainable development, and it operates like a loop and not in a linear system. Okay, so we have the raw materials as the input, and then the design, production, distribution, consummation, and then we have repair or reuse. Then we have what the residual wastes that can be biodegradable or recyclable. And then we have again the raw materials. Okay, so this is the definition for circular economy. And now the biorefinery concept. So what means biorefinery? maybe some of you work already using this biorefinery. So biorefinery is a refinery that converts biomass into energy and products. So this products can reactives, etc. And why are we now using biomass instead of oil for production, energy, and chemicals? So as you know, it's because the price of the fossil resources, but it's also because of the availability of fossil resources is decreasing. But for me, the most important is for sure the environmental concerns that we are having the consequences right now regarding of climate change, etc. So now I have here an example, and it's something that we are doing. I take an example that we are doing from in our lab. How we are doing biorefinery, and the example that I have, it's with algae, red algae. So it's a species that we have here in the coast. It's the Gelidium corneum. So it's a red algae, and actually here until now, this algae, it's captured only to extract agar. So agar-agar, it's also a polysaccharide that it's composed of aragose and agaropeptin. And it's very important for foods as a food additive to do the new gelatin. It's a we can have gelatin without being with the vegetal origin and not animal origin. And until now, actually, they use it to extract this polymer, biopolymer, so agar-agar. And the fisher, the association of the fishermen, the local fishermen, they contact me and they ask me, "Oh, Susanna, do you think that it's possible to valorize more our algae? Because we want to change the economic value or this algae." And I told them, okay, I think so, because usually in algae we have a lot of compounds that are important, not only for food, but for cosmetic and other kind of sectors. And so, but I need to study it. So we study first the composition of the algae. And once we know about the composition of the algae, so we start to develop this holistic approach in cascade that we call biorefinery. So this means that with one sample of algae, we are not extracting only one polysaccharide, but we are trying to extract with the same different kind of compounds that we can find in the algae. So in the beginning, we were able to valorize 2 products with this algae. So 2 products because it was agar-agar, but then the residues from or the byproducts from this algae, it was used as nutrient for the agriculture. But now we are valorizing it more because we have more than 2 products. And I will show you how we did it. So you see, as I told you, so this is the algae, Gelidium corneum, and we saw that we have proteins that we can valorize, different polysaccharides, and also small molecules that have biological activity. So we start by see because the thing is that when you are doing extraction, so in this biorefinery, for instance, if we extract first agar-agar and then the small molecules or proteins, we are degrading the proteins because the proteins, if we have temperatures more than 40 degrees, they can denaturate, as I told you in the first course. And the thing is that to extract agar-agar, we need to use very strong basic solutions and very high temperatures, around 90 degrees Celsius. So the idea it was to start by extracting the small molecules and the proteins before. So in our algae, so around 12% of the compounds it's agar. And so from this agar, we can also separate the two biopolymers, agarose and the agaropeptin from this the agar-agar, sorry, and this polymer it's really have a low sulfur content, so it's very valuable for the kind of applications that we are expecting. And what it was also interesting is, you know, this kind of the algae, they also have cellulose. So we were also able to extract cellulose until now 40%, but now we are improving our method to extract, and now we have more cellulose, because if you see this is a student that did this very nice image of this algae. You see the algae, it's very flexible because we have a lot of variations in water, but we know also, so this is what gives this flexibility. It's the agar-agar, so this polymer, but it's also rigid because it's in a rock. So you see this part of the algae, we have also cellulose. So we extract the different polysaccharides, so including cellulose, and you can see we can even to do some papers from our cellulose. And we also extract the proteins. We have a lot of proteins in this algae. So 18% of the components of this algae, it's proteins. And then we have small quantities, but very important, about these molecules that present antioxidants, and also they can absorb the UV radiation. Tomorrow I will talk with you more about this compound. And so what I would like to show you here, it's that we were able to extract the mycosporins, or these small molecules, the agar, the proteins, some lipids, and then other, so the cellulose. And at the end, we still having some residues, and this it was for instance, to be valuable in fertilizants. So you can see this is the concept of biorefinery. So you have a sample of something, and you can valorize the most compound that's possible that you have in our algae during the extraction. Okay, so talking about the extraction of some compounds from algae, I have again a video to show you, and then I will discuss about my research group. Okay, this is okay. So yeah, it's about pesticides, natural. It's okay, can you see it?
Des pesticides naturels issus des océans. Les algues et les microalgues ont colonisé les océans il y a des milliards d'années.
Oh, sorry. I can't have the subtitles. What we can do, and sorry for the publicity from YouTube. What we can do, I think it's in the list that I send you for this afternoon, okay? And you can see. So it's how can we extract pesticides from algae, okay? Sorry. But in the list that I send you, the subtitles, you have the subtitles, okay? Sorry. Okay. Okay, so now I would like to present my research group. These are the people. Can you hear me?
Yes, ma'am.
Thank you. So this is, they are the people that are working now with me. They are 3 PhD that are finishing in December or defending the thesis in December. And in January, we have new people coming because we had some projects. My research group calls MANTA. So this that comes from marine materials. So and we work. Let me see. Yeah. So the aim of our research group is to valorize marine bioresources, as just I show you, for instance, with the algae, and also to do new multifunctional and sustainable materials from marine biopolymers by being inspired by the function, the structure, or the system of this marine biological materials or marine organisms. Okay. So the main objective is, as I told you, so the development of bio-inspired and sustainable biomaterials to lower the impact of the polymeric materials in marine environment. So we are doing it by taking two approaches. So one is by using, as I told you, marine wastes or fishing wastes. So marine organisms as a source of new chemicals and new biomolecules. And then by being inspired by these organisms. So here I have, for instance, two images. One is we are inspired, and I will show you tomorrow, by the mucus of fish, by the biological activity of fish mucus. And we are also interested, as I told you, by the composites, this biological material and composite material that it's made, the exoskeleton of crustacea and insects. So here it's our mission that I just told you, and the approach. So we look at marine organisms and systems through the eyes of material scientists, because we are so now we also have some biologists working with us, but most of my students and PhD and postdocs, we are physical-chemical chemists. We are chemists. So the thing is that there are already a lot of work about biological materials, for sure, but with the eyes of some biologists, and now we want to have the eyes of material scientists to see how to do new materials. And then it's what I told you. So we explore marine biopolymers, and we used bioinspiration to do our materials. The kind of applications that we are working, it's for biomedical and the cosmetic. For packaging, we have only one project. It's more on cosmetic and biomedical applications. So to do it, actually, we have 4 main research lines in our group. So the first one is because we do materials, but we also extract the biopolymers and the active biomolecules. So we have local, usually it's local residues or byproducts or wastes. And then we develop green processes to extract these biomolecules and biopolymers. So this is the first research line. And the second research line, it's about the making materials, so through bioinspiration, so biomaterials, bioplastics, hydrogels, 3D scaffolds, etc. And then the other two research lines, one, because you see, to show you and to explain our research, I also did it in a loop, because for us it was not also important to extract and to do materials, but also for us it's important to see the impact of these materials in human health. So here in the lab we don't have this the way to do it, because we can't work with human and animal cells in our lab. It's a chemical lab, but we are trying to have this authorization. So we have different kind of collaborations to do to see the impact of our materials in human or animal cells. And the other thing that we study here in the lab, because we also have a big group working on environmental impacts. So what we do, we see what is the impact of our materials in the marine environment. So here we have a little bit more detail about the sources and some of our papers. So about the sources that we have. So we have marine animals, wastes. We don't work with directly with animals. We work with wastes. We don't receive the animals here in the lab. We work with marine plants and algae. And so we try to develop purification methods to have the pure molecules and extraction methods. And here there are some polysaccharides or proteins that we are interested on. And the way the assisted extraction is chemical and enzymatic extraction, microwaves, and we also have a collaboration with someone here at the university with electrical discharges. So this is good because it's a physical way to disrupt the cell walls of our algae, for instance, and to extract better, so the small molecules, and then to extract actually we do it as a pre-treatment. Because for the biorefinery thing, you know, with these discharges, we disrupt the cell wall of the algae. So the small molecules, they go out easily, or they diffuse from the algae. And then it's also easier to extract the polysaccharide later. And then we have, it's like a big institute. So we have a lot of techniques to characterize. We have carbon and hydrogen NMR to do with powder or solid, liquid and gel. We also have a very strong team in analytical chemistry. We have a lot of microscopes and also TGA, DSC, etc. You can characterize all our materials. So then for the development of new bioinspired and sustainable materials. So I have here some materials or some ways that we do to prepare our materials. So assembling routes, chemical and enzymatic modification, cross-linking, and also with electrostatic interactions. Again, the type of the process that we know to do the materials and the kind of materials, and again, the kind of methods that we can characterize all our materials. So regarding the materials degradation, Again, we can do abiotic degradation. We have like an oven that or a simulator of the sun radiation and humidity and temperature that we can actually to simulate the environmental conditions. We can do this kind of degradation to our materials. For the biotic, again, it's a collaboration outside of our institute that we have. And regarding this, we, for instance, we can do it in different kind of waters. So pure water, ocean water, and river or lake water, because, you know, we have different kind of molecules in these different kind of waters, and the degradation can be different, and we already show it with some works that I will show you tomorrow regarding small active small molecules. Okay. And then the research about the impact of human health. So we are interested in different things. So cell protection, tissue regeneration, and since 1 year also drug delivery with the chitosan. So these are our 3 fields of research in regarding the biomedical applications. Okay. And here it's all the institutions that we have collaborations, the people that I'm working with, and some funds that I have, and partners, industrial and public, that I would like to thank to them. And this is our team. So yes, we also receive, because I saw her in the photo, some visiting professors and researchers from other laboratories. So last year we had some a researcher from Brazil, and also from France, working with France. It was working with brown algae, so with the alginate, for instance. Thank you. And then now we can again we can discuss, and if you have some questions. Okay, I just need 1 minute to drink some water. Okay, I am back. I'm back. So we can start our discussion if you like.
Is there any questions? Please ask. I request the students to please interact with professor.
You were able to see to see the subtitles in the video, because I see a comment that it's in French. Yes, it's in French. I think that there are one that it's in English, because this it was a video that it was prepared yes, in French, but you have subtitles, no? It's in English. It's only the last one that it's not possible to see, to have the subtitles in English. I don't know why. But in YouTube, it's, I'm sorry, but yeah, maybe you have a lot of publicity now, but you have the possibility to have the subtitles in English. So, and yes, don't hesitate to contact me and to do some questions by email. And yeah, tomorrow, I will discuss about bio inspiration and biomimetic. um, oh, I like funny questions. You know, it's like this that we learn and we think about. So I was thinking, why nature? So what nature, yes, just take care, attention, because this is also something that my students, sometimes they confused between chitin and chitosan, okay? So chitin, it's the biopolymer that we can extract from the exoskeleton of crustacea, insects, etc., and also from the cell walls of fungi. And chitosan we can also to extract from fungi. So why we have this a still group in so the then if we so this deacetylated this acetylated group in chitin, it's important because I think it's because of this, because for instance, you know, the exoskeleton of the shrimps and crabs. So first, they need this kind of antimicrobial properties, because in water there are a lot of microorganisms, and imagine that they will have like these biofilms with the exopolysaccharides that we learn about this bacteria. If they have it in their all the time in their exoskeleton, it's terrible. So I think it's one of the things because of this anti-microbial activity. And another thing is that we have, so there are an enzyme and that the crustacean that they also have, it's, you know, in crustacea, and it's like the insects, they have what they call the molt. I think it's like this in English. So the exoskeleton, it change every time, you know, and actually the crustacean, the crabs, for instance, they eat this the exoskeleton. So and we have this an enzyme. They have this enzyme that cut the chitin chain in order to degrade it. Okay. And this is also, for instance, taking in account the use, if you need to choose cellulose or chitin to do your materials for biomedical applications, I would say, why not chitin? Because we also have lysozyme, so this enzyme then can degrade chitin. And for instance, for cellulose, we need to inject enzymes that will degrade the cellulose scaffolds. Yeah, it was what I did with cellulose. So I incorporate, but in my case, I incorporate amino groups to have antimicrobial properties. So we did it with cellulose. There are a lot of works on it. You can see it. Thank you for the questions and to be there.
Is there any more questions? If you have any questions, please type in the chat box.
And yes, don't hesitate also to contact me directly, okay? It's the end of the week, you are tired. So the course is until it will finish tomorrow, is that?
Yeah,
So
yeah.
With me, yes, but I don't know if you continue.
Tomorrow is the last class.
Okay,
Yeah.
okay.
5 days.
Mm. Yeah, I agree with you. Biomimicry, yes. Tomorrow we will discuss this a little bit more in detail, but yeah, I'm doing it since now 10 years, I think, and for sure, yeah, by inspiration to look at nature as nature do things, it's really very important. Tony, so I'm doing now, I have a PhD student. It's the first time for drug delivery work, and we are using, so it's a PhD student, it's a collaboration with an university in Spain. So they are biologists, molecular biologists, and so they were looking for a laboratory to do nanoparticles. So we are doing nanoparticles from chitosan that will be serve as drug delivery for the treatment of liver, some disease of liver. So it's about it that we are working, and maybe in 6 months we will have our first publication about this work. We are doing now in vivo studies.
Ma'am, are you doing some work on self-healing polymers?
Sorry?
Ma'am, are you doing some work on self-healing polymers? Self-healing.
Self-healing polymers.
Yeah.
So what can I tell you about it? It's just because it's with an industrial, so I don't know if I can. Let me see. We have some hydrogels that we are developing, but I can't tell you the composition of this hydrogel. Sorry, because it's with an industrial. So, but we also work a little bit on.
Just out of curiosity, when you said about bioinspired materials, I have a few work, I have done a few work on self-healing polymers. So just out of curiosity, I asked.
Yeah.
Is there any more questions?
I was looking at PhD. Yeah. I agree with you that biopolymers, it could be, yeah. Yeah, I think it's good if you do some search about it. What if there are biopolymers, and what kind of biopolymers, and if not, so we can do like something like a comment or a perspective on it. Yeah. Mm.
The first day, I think we have a lot of questions from Rajiv Kumar. He was very interested to ask such interesting questions. Rajiv Kumar. Some
Yeah, for sure. You can contact me for nanogel and microgel also. I have a colleague that works a lot of about it. Yeah, and I think they published something in carbohydrate polymers recently. And I have so this colleague, he opens a startup, a company to produce these kind of things. Thank you. Thank you.
I think there are no more questions, ma'am. Then shall we?
Wait, yes, sorry. Yeah. So thank you for today and see you tomorrow for our last lecture. I think it's yeah, maybe I have for 40 minutes talking and then we can discuss about bioinspired materials. Okay?
Okay, ma'am. Okay. Shall
Okay.
we take a photograph also?
Yeah, for sure.
Ma'am, can you stop sharing? I think.
It's okay?
Can you stop the sharing? No, no.
Oh, sorry. Yeah. For sure. Sorry.
Okay, okay. Please, everyone, can you switch on the camera? Okay, ma'am. Thank you so much.
Okay, thank you. See you tomorrow.
See you, ma'am. See you tomorrow.
Bye.